So a very good morning to everyone. Welcome to our today's webinar, Breakfast at UMHealth, episode number 32. So in 2021. Our speaker for today is Professor Chung Yong. So first of all, I wanted to give a brief introductions for him. So for information, Professor Chung, he is the Professor of Pharmaceutical Sciences and one of the founding members of the Department of Pharmacy at the Faculty of Medicine, which is now the Faculty of Pharmacy of University Malaya. He worked in the UK as a research associate prior to joining University Malaya. He published more than 100 ISI articles and consulted for European or UK multinational and local industrial companies. He was named as top 2% scientists in the world in the subject field medicinal and biomolecular chemistry, and career-long citation impact by Stanford University in 2020. So he will be talking about applications of micro or nanotechnology in medical and pharmaceutical sciences today. So without further ado, the floor is yours, Prof. Chung.
Good morning. I would like to say thank you to UMHealth for giving me the opportunity to share with you some of the work we had carried out and published with our collaborators from the Faculty of Medicine of the University of Malaya, Hoshi University, University of Toyama, and National Chiao Tung University. In this, I shall focus on the applications of micro and nanotechnology in medical and pharmaceutical sciences. Micro and nanotechnology can alter the properties of drugs and the delivery systems to improve drug targeting and to reduce unwanted side effects to ultimately improve the clinical outcome. Similar micro and nanotechnologies can also be applied to biosensing platform to facilitate SARS-CoV-2 research. In this, I shall give two case studies as examples. In case 1, I shall look at pH sensing micro nanoconstructs to improve the delivery of oral insulin, whereas in case 2, I shall look at design and application of a rapid microbiosensor to screen for potential compounds that elevate SARS-CoV-2 cell entry. In this example, I shall illustrate the use of pH sensing micro nanoconstructs as they travel through the gastrointestinal tract to improve the delivery of oral insulin. The usual route is parenteral or subcutaneous, with some obvious disadvantages such as allergy, pain, itching, insulin lipodystrophy, and others. There are other possible alternative routes. These include pulmonary, intranasal, and oral. The main advantage of oral insulin is that it mimics the natural secretion of insulin. As we all know that insulin as a polypeptide is easily denatured by the stomach acid at pH 1 to pH 3, and also the peptide enzymes in the gastrointestinal tract would make it inactive. To overcome this, we apply the concept of putting insulin molecules in a rigid polymeric container, protecting them from being damaged by stomach acid and peptases. However, once the insulin molecules transverse through the gastrointestinal tract at the pH of 6 to 8, the protective rigid polymeric container must start to open up and disintegrate to release the insulin molecules and allow them to be absorbed into the bloodstream. To do this, the rigid polymeric container must be able to sense pH gradient and alter its structure as pH changes in the gastrointestinal tract. We therefore introduce additional carboxylic acid groups to the polymer to mediate these effects, as shown in the diagram on the right-hand side. A lot of us think we use fancy chemistry and materials, but in reality we use materials found in the fish market, such as seaweed and crab or prawn shells. The trick is to put in just enough or the right amount of carboxymethylated groups to carrageenan from the seaweed to give it the ability to sense pH and changes its structure according to pH level. The chitosan from the crab shell is to impart the ability of the nanoparticles to stick to the wall of the gastrointestinal tract to facilitate absorption of insulin molecules into the bloodstream. I'm not going to say anything much regarding the experimental steps, except it suffice to say the work involved extensive instrumentation, mathematical modeling, and in vitro and in vivo experiments. This slide shows how the nanoparticles look like. If you look at the photomicrographs labeled A and B, the dark or black round blot or spot is the nanoparticle constructed of carboxymethylated carrageenan and chitosan, and they contain insulin molecules inside. The diameter of the nanoparticle is around 600 nanometer, containing about 10% of human insulin molecules by weight, and has the ability to adhere to the gastrointestinal tract. We then take these nanoparticles containing insulin and pack them into capsules and test the performance using first in vitro insulin release study in simulated gastric fluid and simulated intestinal fluid with and without enzymes. Second, in vitro insulin transport study using a parallel artificial membrane. Third, cytotoxicity investigation using a cargo 2 cell line, a gastrointestinal cell line. Fourth, to determine the bioavailability of insulin using a diabetic rat model. The figure on the left shows in vitro release of insulin in simulated gastric fluid at pH of about 1 to about pH 3 from 0 to 2 hours, and also simulated intestinal fluid at pH 6 to about 8 from 2 to 12 hours. The vertical axis indicates the level of insulin release, and the horizontal axis shows time. If you look at the red or blue lines, you can see the insulin release from the optimized nanoparticles in the simulated gastric fluid is actually very low, less than 5%. However, as the nanoparticles goes through to simulated intestinal fluid with a pH range from 6 to 8, the insulin release from the same nanoparticles increase to 90% cumulative release at 12 hours. The diagram on the right-hand side shows how an insulin transport study is carried out. In this, we first added the nanoparticles containing insulin to the top part of the well and then measure the movement of the nanoparticles containing insulin across the blue-colored artificial transwell membrane into the lower chamber. The results clearly show there are no passive transport of nanoparticles through the intestinal or artificial membrane. This slide shows how we investigated the transport mechanism of insulin from the nanoparticles across the gastrointestinal tract using cargo-2 cells. Cargo-2 cells are gastrointestinal cells that are often used for gastrointestinal transport studies. If we look at the diagram at the bottom left-hand corner, we first grow a layer of cargo-2 cells on a transwell membrane insert until they are highly confluent without gaps between the cells. That is to say, the junctions between the cells are tight, and at this stage is highly representative of the cells in our gastrointestinal tract. We know this by measuring the electrical resistivity between the cell layer as shown in the diagram. To investigate the transport of insulin, we first added free insulin as a control to the top chamber of the transwell insert above the cargo-2 cell layer and measured the electrical resistance over time. The results are indicated by the light blue line on the top right-hand corner, and the electrical resistance remained high and did not change according to time. We further confirmed the results by measuring the amount of insulin present in the lower chamber of the transwell insert using a human insulin ELISA assay kit. If we look at the top right-hand figure, there was no human insulin detected in the lower chamber of the transwell insert. The combined results clearly suggest free insulin is unable to cross the cargo-2 cell layer or gastrointestinal membrane. However, if you repeat the same experiment using nanoparticles containing human insulin by adding to the top chamber of the transwell insert, the electrical resistance of the cargo-2 cell layer decreased and then recovered against time, as indicated by the dark blue line on the top left-hand figure. There's also corresponding increase of human insulin against time in the lower chamber of the transwell insert, as indicated by the dark blue line on the top right-hand figure. What do all this mean? The mechanism of action is summarized by the diagram on the bottom right-hand corner. The results here clearly show when the chitosan carboxymethylated carrageenan nanoparticles adhere or stick to the cargo-2 cell layer, they have the ability to transiently open up the tight gap junction to give a transient drop in electrical resistance. The temporary opening of the tight gap junction of the cargo-2 cell layer allowed the release of human insulin molecules from the nanoparticles to go through the transiently opened gap junctions between the cells to be absorbed into the bloodstream. This mode of insulin transport is known as a paracellular transport. This scheme here basically shows how we conducted the in vivo hypoglycemic study using streptosin-induced type 1 diabetic rats. We carry out oral feeding of nanoparticles containing human insulin in capsules to the rats and measure the changes in blood glucose and also measure the amount of human insulin present in the plasma of the rats using a human insulin ELISA kit. The figures show the hypoglycemic effect and serum human insulin concentration in diabetic rats. The figure on the left side shows hypoglycemic effect against time. The vertical axis is hypoglycemic effect, while the horizontal axis is time. Here, at time equal to 0, we fed the rats with nanoparticles with insulin, empty nanoparticles, and oral insulin solution. We also include subcutaneous injection of insulin as a comparator, as it is a normal administration route. First, empty nanoparticles, empty capsules, and oral insulin solutions did not give hypoglycemic effect. Second, subcutaneous injection of insulin gave a rapid and shorter hypoglycemic effect. Third, nanoparticles containing insulin gave a dose-dependent and prolonged hypoglycemic effect. The data were corroborated by the right-hand figure, where we measure the human insulin present in the serum of the rats. First, empty nanoparticles, empty capsules, and oral insulin solutions did not give any measurable human insulin in the serum. Second, subcutaneous injection of insulin gave a rapid and shorter duration of insulin level in serum. Third, nanoparticles containing insulin gave a dose-dependent and prolonged level of insulin in serum. The results suggest insulin entrapped in the chitosan carboxymethylated carrageenan was released in the gastrointestinal tract and absorbed into the bloodstream of the rats to give increase in serum insulin and hence hypoglycemic effect. We then relooked at the data and calculated the bioavailability of nanoparticles containing insulin versus insulin injected by subcutaneous route. The data in the bottom table suggests the bioavailability of nanoparticles containing insulin via oral route is only about 16% to that of the subcutaneous injection. Can we improve its bioavailability? Can we use the bioavailability data obtained in rodents or rats and make some prediction in humans? There are two main important studies looking at hundreds of smaller studies comparing the differences between bioavailability of drugs or peptide drugs in rodents, rabbits, primates, and humans. The studies summarize that first, rat intestinal absorption of drugs is similar to that of human intestine. Second, the authors gave a correlation equation between rats and humans. Using this approach, we suggest the predicted human bioavailability is about 47% or 3-fold higher compared to that of experimental rat bioavailability. In theory, you probably lose about 50% of the insulin via oral route. The main advantage is that this route mimics the natural insulin release in the body without the sharp spikes and appears to be useful for basal insulin control. In summary, first, insulin can be delivered orally if we apply micro or nanotechnology to the delivery system to protect the insulin molecules when they transverse through the gastrointestinal tract. Second, despite the bioavailability is only 16% relative to the subcutaneous route in rat, but the predicted human bioavailability is about 47%. Third, oral insulin delivery mimics the natural secretion of insulin with less spikes and could be useful for basal control. We shall now talk about something completely different, something which is more current. In the second case here, we shall look at the design and application of a rapid microbiosensor to screen for potential compounds that alleviate SARS-CoV-2 cell entry. This work was started in March last year, 2020. In any biosensor work, such as the pH meter and probe, there are three key components. First, biomolecular interaction, that's to say, interactions of the molecules you are trying to measure with a probe. Second, the transducer or probes, which converts the degree of interactions to an electrical signal. Third, the sensing system to register or measure the intensity or degree of interaction. This figure shows we first make a palladium thin film electrode, then coated a layer of recombinant ACE2 or angiotensin converting enzyme 2 on the surface of the palladium electrode. We then add a recombinant SARS-CoV-2 spike protein or S protein to study the interaction between the immobilized ACE2 and spike protein. It is believed that the interaction of SARS-CoV-2 spike protein with cell surface ACE2 promote the SARS-CoV-2 cell entry. At this point, we also realize the possibility of using this system to investigate potential compounds that may increase or inhibit ACE2 SARS-CoV-2 spike protein interaction. This slide shows our SARS-CoV-2 biosensor can recognize pseudovirus and S protein. First, dose-dependent increase of electrochemical impedance signals related to the binding of SARS-CoV-2 pseudovirus to the ACE2-coated electrode. Second, a dose-response curve representing the S protein concentration dependent interaction with ACE2 generated by the biosensor. We realize the system is capable of screening of drugs that affects the SARS-CoV-2 S protein ACE2 binding. It's able to identify S protein ACE2 binding positive modulators and inhibitors. We actually managed to identify two main groups of ACE inhibitors for the treatment of hypertension. For S-protein ACE2 binding inhibitors, we have perintopril, perintoprolate, ramipril, ramiprolate, inhibit S-protein ACE2 binding by about 42 to 72%. What does this mean? Essentially, the drugs here inhibit the binding of S-protein to ACE2 to reduce the S-protein cell entry. Whereas for S-protein ACE2 binding enhancers, we had enalapril, enalaprilate, lisinopril. They increase S-protein ACE2 binding by 24, 174 and 67 folds. The results clearly show these enhancing drugs promote S-protein ACE2 binding, that's to say, to increase S-protein cell entry, or probably to increase COVID-2 or SARS-CoV-2 infectivity. This slide shows the effects of positive modulators and inhibitors on SARS-CoV-2 infectivity to the ACE2 expressing OEC-M1 cells. If we look at the left-hand side of the slide, we demonstrated that quantum dot S protein can be taken up by surface ACE2 expressed on OEC-M1 cells using confocal microscopy. We then look at the infectivity of SARS-CoV-2 in the presence of these drugs and demonstrated that enalapril, enalaprilate, lisinopril increase SARS-CoV-2 infectivity, whereas perintopril, perintoprolate, ramipril, and ramiprolate decrease SARS-CoV-2 infectivity. The results here corroborated with our earlier biosensing data. This slide probably explains why some ACE inhibitors block ACE2 S-protein binding and others promote the binding. Although the number of compounds tested is rather small, but the trend suggests the presence of a bulky rigid fused ring structure in ACE inhibitors block ACE2 S-protein binding. The bulky rigid ring structures in perintopril, perintoprolate, ramipril, and ramiprolate are labeled A and B, whereas the corresponding single ring structures in enalapril, enalaprolate, lisinopril, and captopril are labeled C. This summarizes the significant points of findings in this work. First, the biosensor platform can be used to screen for drugs that may inhibit and/or increase SARS-CoV-2 infectivity. Second, the same platform may be used for other emergency infectious disease sensing or detection. These are some of the references we use for this presentation. Thank you for listening. Before I end, I would like to thank my collaborators in University Malaya, Hoshi, and Toyama University in Japan, and also National Yang Ming Chiao Tung University in Taiwan. Thank you very much.
Thank you. a very interesting presentation. So sorry for the technical hiccup just now. So now I would like to open the sessions to questions and answer. So for information, you can see that there is a Q&A icon beside your chat functions in your Zoom. So you can actually click that to type in your questions. So we have already one questions directed to Prof. Chung. So Prof. Chung, you want to answer it?
Okay. Okay, Dr. Heh, thank you. The questions on what would clinical situations like diarrhea affect bioavailability? If so, would it affect it? I presume, with diarrhea or even with food, it would definitely affect to a certain extent of the bioavailability. Then the second part, I think, is if so, this variable absorption may affect our diabetic management of brittle diabetics and may also put them potentially with low level of blood glucose if they are not closely monitored. I suppose, it would actually, if you have diarrhea, it would affect it definitely. And sorry, I'm not very familiar with what the brittle diabetics, so I can't comment on that. Okay. So.
Okay. So thank you, Prof. Chung. So you are welcome to put in any questions. So maybe I can ask questions myself. So how long it takes for you to achieve all this work, actually, Prof. Chung?
For the oral insulin, we were lucky enough to have a few grants to support the work, but it has taken quite a few years. Okay, so I would say it would be at least about 6 years. Okay.
Okay, so there are questions coming in. The first one is brittle diabetics are those who have huge sensitivity or poor control of diabetes mellitus. That is the explanations for the previous one. Would you want to comment on this?
Sorry, I'm not very familiar with this part. But I suppose, those with very poor control, I think it would be good to actually monitor your glucose level.
Okay.
Okay.
So how about the next question is, what was the cost incurred for this platform? I think maybe for both of the study that you mentioned.
If we are talking about just the research side of it, okay, at least for the first one, it would have cost us probably around 400,000 or so. Okay. Now, but if you want to develop it further and actually to see whether you can actually sort of use it clinically, then what we need to do is actually to demonstrate the bioavailability is actually much higher than 16%, i.e. it has to be, in human, it has to be at least about 50% for it to be more or less slightly more cost effective. So to do the sort of clinical trial, it would cost a lot of money. So I suppose, perhaps some of the clinicians may be interested in this, can actually take it forward. Okay. Then for the second one, now, because it's a collaboration actually with our Taiwanese group, most of the financial aspect is actually from the Taiwanese side. So I'm unable to give you the figure for that, but it will not be cheap because we actually needed to get to make the recombinant proteins and so on. So those would actually cost money, especially making the S protein, making the ACE2. So those would cost quite a bit, I think. Okay. So.
Okay. The next question is, how long does it take to create this the platform? So, and then in terms of COVID-19 detection, has any study been done so far in human samples?
For this particular one, the initial part, the pilot part of the work, the prelim work actually took at least about, well, slightly less than a year, about a year or so lah. Okay, about a year. Then, in terms of whether we have done it in human samples, as far as I know, at least we have not been doing it in Malaysia, but it is possible that our collaborators have tried in with human samples. Okay, there's a possibility.
Okay, thank you for the answer. The next question is, how fast or easy to use the biosensor to screen potential drug or small molecule to block the binding of ACE2 and SARS-CoV-2?
Okay, for this one, the volume required is actually very small. It's actually in microliters, but it will take at least about 10 to 15 minutes or so for you to actually get the complete reaction. So to actually generate the whole curve, if you have at least 4 points, okay, it will take you at least about an hour or so. If it's 8 points, then it would be at least about 2 hours or 3 hours. Okay. So it's not something like you put it in now, you get the results. No, you have to actually allow time for the reaction to go into equilibrium and so on. And then you check the endpoint. So altogether, to have a dose response of about 8 points, it would take about 2-3 hours or so.
That's pretty good already, 2-3 hours. So maybe I can ask, is there any other potential candidates that you or your collaborators have screened so far? Maybe you don't need to say what is the compound, it's confidential, but maybe do you have any other potential candidates?
We have, we are actually sort of, we haven't actually done much in Malaysia because most of the thing is actually like the recombinant things is actually from our collaborators. Okay, so it is possible that the industry, at least their collaborators, actually also screen into a few other compounds. Okay, as I mentioned earlier, the compounds we tested for the paper is actually quite limited. So in order to actually have more confidence over this, we needed to have a larger pool of samples. Okay, thank you.
Okay. So I believe other people might be interested to know that whether this platform it's also can detect like compounds for the mutant to inhibit the mutant.
Okay. It's a possibility. All you need to do is actually change the use of what mutant S protein. So as far as I know, there are some companies actually making the recombinant mutant protein. But I mean, to do this sort of work, it costs money. So is there anybody interested to continue on this? So if anyone is interested, we can actually look into what the mutant S protein.
For those who are interested, you can contact Prof. Chung. Okay, so any more questions? So I think there's no more question coming in. Okay. So maybe before we end this session, I would just want to ask Prof. Chung to give like what is your for information for all of your information. Prof. Chung says will be retired in 3 days, if I'm not mistaken, 21st of August. So he will become an honorary professor after this in the Department of Pharmaceutical Chemistry in the Faculty of Pharmacy. So that's why. What is your the one words you want to send to all of us? Based on all your achievement until now.
I think I actually joined the faculty, initially Faculty of Medicine, or the Department of Pharmacy within Faculty of Medicine in 1995, when it was actually under Prof. Anwar Zaini. Okay, so I mean, throughout the years, we were lucky enough to actually had support from all the deans, the university, to manage to secure some funding to do research. And we were very lucky to actually have money from HIR, the high impact research, for part of the insulin work and so on. And before that, it was actually from the university research grants, I think. So I think the most important thing is try your best. And in terms of research, once we promise what we want to do, try to actually carry out and complete the all or most of the objective proposed in the grant proposal. Basically, just do the best you can. Okay, so I mean, we are unable to compete with what European or the Americans in terms of what facilities and so on. So it is also important to actually have networking, have collaboration with overseas partners. So this will actually help us in terms of research, especially we don't have some of the fancy equipment which we need in our work. So perhaps they can actually provide that. So with my years in we actually have collaboration with Texas A&M, then Spain, UK, Serbia, and Turkey, plus a few other places as well. Not forgetting Taiwan, China, and Japan. So it is important to have international and national networking. So thank you.
Okay, thank you for the tips, Prof. Chung. Okay, so with this, I would like to thank all of the participants. So I think we will end our session for today's breakfast at UMHealth. Yeah, thank you very much. Thank you for joining. Yep.
Thank you.
Yep. See you next week.